Hi, this is Miss Litton, and this is 7th period support for AP Biology, Chapter 14, Biotechnology and Genomics. Say hi.
Hi.
Right, and we are going muy rápido. Okay, so the first thing we did is we just talked about just generic different kinds of cloning, okay? But then that brought us to the idea of cloning a gene. And you could clone a gene, you could, because you might be wanting to get a product from that gene, you might be trying to analyze the base sequence of that gene, you might want to alter an organism's phenotype. So we needed to start talking about the tools of a genetic engineer. So two big ones are anything to do with recombinant DNA technology, and for that we need to know about REs. What are REs? And ligase, right? So those would be two key things to know about recombinant DNA technology. Restriction enzymes are named restriction enzymes because they were first isolated from bacteria trying to defend themselves against what? Viruses. And so they would restrict the growth of those viruses by cutting up the viral DNA or the viral RNA, whatever it was. So we use restriction enzymes to cut DNA at a specific series of bases. And we create a staggered cut. We come in and cut across and come out in order to create what? Sticky ends. Sticky ends, which are then make it so we can mix and match DNA from different organisms, or the same organism, whatever we're after. Now, we know that it'll form the hydrogen bonds to bond them back together, but what seals the phosphate sugar backbone? The ligase. So those are the two enzymes we need here. For the polymerase chain reaction, what is the polymerase referring to? DNA polymerase. And basically, when you do the polymerase chain reaction, it requires a machine. What is the machine that it requires? A thermocycler. And it's called a thermocycler because you are cycling the what? Temperature up and down. The up is for what reason? Why would you increase the temperature? To denature the DNA to go from being double-stranded to single-stranded. You don't want to have an enzyme do that, otherwise they'd never stick, right? You wouldn't be able to make new DNA. So you do it physically with temperature. You separate the DNA strands. Then you need to use primers to identify where you want to replicate, what portion you want to replicate it from. What would that be like when we talk about CRISPR? The guide RNAs, remember that? Remember that? Those were cool. Okay, so you need to have a little primer to mark where you want it to go, and then you need some DNA polymerase to make that copy, right? And now you've gone from one copy to 2 copies. And then you go back, you go from that cooled state where you can do that, where you can do the replication, you heat it back up again in order to make it single-stranded. So you have to use an enzyme, a DNA polymerase that can withstand that heating and cooling, heating and cooling. That's why we use what? TAC, because that comes from who? Thermus aquaticus, who is under what domain? Archaea. All right? And so that allows us to go to that hot cold. Now, we know how to cut and splice DNA, but we might want to check our work, right? Like an answer key. What is something we can do to check our work? Gel electrophoresis, which will sort DNA according to its size. Well, you could just say, well, I have weird sized DNA. You have the right size DNA if you have spliced the right genes together and you know where the recognition sequences are, because remember, restriction enzymes cut a specific series of bases. Yes? Called the recognition sequence. So if our recognition sequences are in the right place, then we'll be able to cut that DNA in the right location, and then we have an RFLP. Do you remember what that is? Oh, go away. Do you remember what an RFLP is? Yes, RFLP. Restriction fragment length polymorphism. Restriction for cutting. We cut the DNA into fragments of different lengths. Polymorphism, there's many different sizes, and that would be specific to that individual. That's like your DNA fingerprint. Okay, so that was a lot, I know, that I just did right there on that page, but I was hoping you would get it. Okay, so here you can see the restriction enzymes cutting, and you can see the overhangs, and the idea is if you cut with the same restriction enzyme, then you will have the same overhangs, the sticky ends match, so that enables you to splice the DNA back together using some sort of ligase. When we want to splice in a eukaryotic gene into a prokaryotic cell, though, we have to remember that eukaryotic genes have introns, but prokaryotic genome does not. So we can't give them any introns. So we have to have a clean copy, just exon, exon, exon of what we want. So how can we do that? Instead of starting with the DNA, what do we start with? RNA. What kind of RNA? mRNA. Yes, mature mRNA that's already had cut, tail, and snurped out the intron, or cut out the introns. So then you use that RNA and you work backwards using what? Reverse transcriptase, which then makes a clean copy of that DNA that we can then put into the prokaryotic, either a plasmid or its chromosome. But we need to make sure we have a way to switch it on now, to turn that gene on. Otherwise that gene will be silent. If you put a gene in next to it and there's nothing to turn it on, it will never get transcribed or translated. So what do we use? What would we have to put it next to? A regulatory region. What would be the regulatory region? The operon, right? What part? The operon refers to the whole thing, including the genes, the structural genes. What do you mean specifically? The operon is everything. It's the full unit.
The operator.
Yeah. RNA polymerase binds to the promoter, right? So you have to have it next to a promoter that you can turn on in some way. And that's going back to chapter 13. Okay? And then your vector is either bacterial, plasmid usually, or a virus. Okay, now as that is settled into your brain, do you have questions? Ask your bio buddy, do you have questions? Okay, come back to me, Go ahead.
So bacteria...
I can't hear.
Bacteria can't cut and splice introns and extrons. So if you had an intron there, could it express it in the wrong way?
Yeah,
Okay.
exactly. It would just transcribe it like it was. It wouldn't know intron, exon. It would just know DNA, and it would just keep transcribing it. So probably you wouldn't get your final product. The other thing you have to worry about, though, is for us, especially as eukaryotes, is our proteins have to be folded into the right shape, right? What do you need for that? Yeah, so you could have a way to turn it on, but if it has some special folding pattern and you don't have the chaperone proteins getting translated as well, you're not going to get folded in the right way. Okay? All right. So then we talked about PCR already. Now let's talk a little bit about gel electrophoresis. Remember, gel, we think gelatin, so it's a gelatin that you're putting it into. Electro, meaning you're applying what? Electricity to it. Gel electrophoresis. And it's going to sort the DNA according to its size. The smaller pieces will move faster through the gel because they can get more easily drawn through that carbohydrate-like mesh, all those sugars in there. An exception to that is you could have a whole plasmid. I'm just telling you this because I don't want to tell you any lies. You could have a whole plasmid that supercoils like this and becomes really streamlined, and it will shoot right through. And we're going to see that later in our lab, so that's why I'm bringing it up now, because those can move through pretty quickly. So you load the DNA in, turn on electric current, sort it according to size, and then you compare the banding patterns, and you can, what you would have predicted. You can have a control. And you will have that in your lab too, like you'll have a tube of a bunch of 10,000 base pair fragments and 8,000 and 6,000, 4,000, 4,500, all the way down to 500 base pairs. So you know that's what's in your control tube. So you will compare your fragments that you see to the control fragments to know how big it is. And so you can verify then that you've cut the DNA correctly. All right, we talked about mitochondrial DNA, and the importance of mitochondrial DNA, the little twist on that, is that it only comes from your mother. And so you could trace just your mother's lineage back by just analyzing your mother's DNA. And then we jumped into all the ways you can use these tools now that you understand them. And we talked a lot about genetically engineering bacteria. You could make them so they make different products. Right now, there was an article or something on TV last night where they're getting bacteria to make the spider web protein. And the strength of a spider web, that protein is so much stronger than just like regular rope that we make from trees or whatever, our synthetic rope. So you can get it to break down oil. You can protect a plant from frost. You can get it to make human proteins that we need, clotting factors, antibodies, all sorts of things. So you genetically engineer the bacteria to do that. And then you just lance open the bacteria and harvest your product. You can genetically engineer plants. To do that, you're going to have to get that DNA inside that plant. It's a little bit, you will put it inside a bacteria, so you will have experience at this. But to put DNA inside of plants, you have to get them at that embryonic stage, and you have to remove all their cell walls so it's more likely to occur. So you can genetically engineer plants. There's that one. There's this one. Okay, genetically engineering an animal, a little more tricksy, because you're going to need to get it into the egg. You get it into the egg, unless you're using CRISPR technology, you can get it into the egg, and then every cell will have that DNA in it. So you can micro-inject it in, and then you would, once you've formed, that's going to be kind of tricky, make your transgenic goat, then you're going to want more of that, and so you'll have to take the nuclei out of those cells and put them into enucleated eggs that have their nucleus removed so you can get more clones, et cetera, because you won't have them reproduce through sexual reproduction. You would want to take a diploid nucleus from this cloned goat and put it into an egg as a diploid nucleus. So transfer the nucleus only. Did you understand what I just said there? What you would do is, let's say you have your goat, your cloned goat that you like. You genetically engineered him, you love him. Okay? You love him. Okay? It's important. If you don't love it, then it doesn't work. Then you take some other goat and get him to donate eggs, or her, to donate eggs, and you enucleate the eggs, meaning you remove the nucleus, which would be haploid because it's an egg, right? You remove the haploid egg nucleus and take a nucleus from your goat that you formed and put it into that egg. Now do you have it? So I take it from my genetically engineered goat, I take one of his nuclei out and replace the nucleus of the egg with that diploid one. And do that, and then I put these into some host uteri, and then you then have your more genetically engineered goat. Yes?
When you replace the nuclei, it still has a different, the clone still has a different mitochondrial DNA, right?
Yes. It will have the mitochondrial DNA of that donor goat, right? Yeah, that part will, yeah. So it's kind of a little, Frankensteinish, but cool. Yeah. You can use that as an extra on your English when you write. Thank you. Okay. Now, there should be a lot of extras you can pull from biology for your, yeah?
Simultaneous.
How was Malthus?
Oh yeah. It helped me. I think
Did it help you?
It was our essay question.
It was our essay question.
Oh yeah, that was good.
We had it last year.
Awesome. Okay. Let's keep going. There was gene therapy, in vivo and ex vivo. Which one is it if you remove some of my bone marrow, fix them with a virus, and return them to me? Ex vivo. Okay, if you give me I sniff something in like a virus or something and it goes in through my lungs, then it would be in vivo. I don't remove them. Okay, perfect. We started talking about genomics. The biggest thing on genomics is it used to just be central dogma. DNA codes for RNA, which codes for proteins. But now we know that's not it. That's just not it. Because when we look at our human genome, what percentage of our human genome do we actually transcribe and translate into a protein? 1%. Yeah, 1%. Okay? So there's so many other things. We have intragenic segments and intergenic. What are intra? Within the gene. Intergenic is outside of the gene. And those can sometimes exert what? Yeah, it looks like dominance. Control. right? They could be have an aspect in whether that gene is transcribed or translated. And then we know about epigenomics, right? We have to look at the things external to the gene, attached to a histone or attached to the DNA that influence whether that gene is transcribed or translated or not. Looking at post-translational modifications of a sequence of amino acids to what protein it'll finally be and how much of that you have. So we know we expand our idea of the human genome, and we can look for small differences, single nucleotide polymorphisms within individuals that could cause no change or tremendous changes amongst individuals. Here's your intergenic. If you had a section in there, it would be intra. In that case, it would probably be a what? Intron, right? Introns and exons. Remember that? Okay. We looked at CRISPR and that Cas9 are like scissors. Cas9. I can't think of any connection for you. I have nothing.
Castrate.
Anybody have anything? Castrate. I like it. To castrate is to cut. Nice. He screams. Castrate. I'm so glad this is getting recorded. Castrate. So you're going to remember that. Cas9 is scissors. And then this is easy. Guide RNA, right? We must be tired. Yes? I know, but we already used Casper the ghost with the other one. P53.
Those are caspase.
Yeah, we're castrating, and he's going, nice. All right. So come back to me. It's Friday. It's clearly Friday. So we can use it to edit genes, but remember they even used the Cas9 where they disabled the scissors so it didn't cut anymore, but it just brought something else with it, like a transcription factor or a repressor to control the way that gene was working. Or they would cut it knowing the cell would probably repair it poorly because they just sliced it like this, and they know it repairs poorly, and then they can turn off that gene as a result of that cut and that repair. So we looked at that. What was this? All right, I talked to you about that already. And we looked at genetic profiles. Remember the guy on the slide, and he had all 30,000 genes, and he said, you can see here the 30,000. Okay. Okay. So where they can, but the point is, they can see if you have a correct copy or a incorrect copy of a gene, to know more about you, and or might know how some drug treatment might impact you in looking for patterns. We looked at proteomics, where you're looking at the importance of just the way the remember we have more proteins than we have genes that code for them, right? And that's because of exon recombination or post-translational modifications. They give us a variety of proteins from a smaller number of genes. Bioinformatics, that's using a computer, but we're talking about massive amounts of data that you're processing from a lot of different, statistics in order to better serve whatever maybe that drug is, or trying to enhance what you're already doing. Expansion of genomics, we already talked about that. We look beyond just the genes, but we look at the information in between the genes and looking at those proteins they code for. And that, my friends, was it.
Can we do some questions?
Sure.
What should we go in
What did you say?
Can you say
That was for period 5. We already did that.
Period 5.
We did
Yeah.
that already.
Yeah, we can do some questions in here. You see what I have? Here's these. Why don't you go in as something fun you're looking forward to this weekend?
The joke is, the German.
I didn't get them.
Yeah, no, that was the joke. The castrating is German.
What's Bra's sound effect too?
Wait, what are we going to ask?
Something fun to do this weekend.
Studying for bio.
No APBio.
Dance, sleep.
Oh, what's the 19th?
Bozeman. Can't wait to get me some Bozeman. Yes, you have to study for the final. How many days away is it? Like 17 days away?
No, not
Yes?
Watching 12 hours both when Cal was studying for the final.
Okay, what kind of studying is that? Input studying. You also need to do output. Yeah, I recommend you start with output. Use that to analyze what you know and what you don't know. Then use your input on what you really need to improve upon, and then do output all over again and see if you've learned anything. Do you understand that? I would want to save time. You can start reviewing your essays. Essay. Crying while studying like heck. Does anybody want a piano or 2 bookshelves or a dining room table?
Oh, yeah, I have a
chairs.
Chairs?
Yeah, dining room table, 6 chairs. Nobody has claimed it yet. A piano and a piano? No,
It's number 2.
Yeah, must be. One new one. 5, 4, 3, 2.
What's your
Was it only B?
It was A and D. A and D. I didn't think these are the only 2 I didn't give you in class. Okay, all the other ones you have done in class, I encourage you to look at them again because you probably have them in your group shared notes, yes? Okay, I have to go to appointment, so love you very much. You're awesome. Have good weekends. Have some toast if you get tired. Maybe a little peanut butter on top.